Our protocol is significant because it can be used to identify genomic sequences that cannot be isolated from co-purifying sequences, which may themselves only be partially known. The main advantages of this technique is that it's inexpensive, using mostly free software that can be downloaded, as well as flexible. You can apply it to many biological questions.
Potential applications include identifying bacterial vaccine targets and identifying viruses whose sequences are extremely different from known microbes. This method can be applied to any system in which the unknown cannot be experimentally separated, as long as a reference sequence without the genomic target is available.
This technique takes some trial and error, so patience is important. You may need to troubleshoot the programs. You may use programs different from the ones we describe here. Use the user manuals as much as possible. Visual demonstration of this method is helpful because computational work is dependent on a basic understanding of how command line programming is structured.
To begin, use Trimomatic 0.32 to remove Illumina adapters and low-quality bases. Use Pair version 0.9.11 to create high-quality merged reads from Trimomatic output paired reads using default parameters. Then use Reptile version 1.1 to error-correct the reads, produced through Pair. Finally, use Trinity version 2.4.0 in default mode to assemble the corrected sequences. For strand-specific libraries, use the -ss_lib_type parameter. The output is a FASTA file that will be placed in a new directory called Trinity_out.